Bom dia a todos! E cá estamos no segundo dia deste 19º encontro Congresso Ibérico de Entomologia e estamos aqui reunidos para a segunda palestra plenária, que promete muito. E antes de darmos a palavra ao nosso orador, eu tenho aqui algumas recomendações para quem nos está a ouvir. Eu creio que uma grande maior parte dos participantes estão a ouvir-nos no Opin e eu vou começar por, se calhar, fazer aqui algumas chamadas de atenção que podem ser úteis. Para já, atenção que o nosso programa está sempre no horário de Lisboa, mas eu sei que muito afortunadamente estão-nos a ouvir de vários sítios do mundo e o Opin aparece-vos no vosso fuso horário, portanto têm que ter o cuidado de fazer a verificação que horas estão em Lisboa, porque é a hora que nós estamos sempre a funcionar, ok? Depois aqui algumas chamadas de atenção. Nós temos muitas possibilidades de interação aqui no Opin, portanto, gostava muito que as aproveitassem. Portanto, se vocês virem aí no vosso ecrã, veem que na base esquerda, na lateral esquerda, do lado esquerdo, tem um menu. Nós agora estamos aqui todos no palco, mas se descerem tem as sessões e nessas sessões vão ocorrer, nesse menu de sessões, vão ocorrer todas as sessões paralelas de comunicações orais. Mas se entrarem aí, além das sessões paralelas, têm também uma sala de suporte técnico, onde podem ir sempre, está aberto em permanência, e podem ir aí tirar dúvidas, como é que partilham a tela, como é que querem fazer alguma coisa que não conseguem, têm dúvidas, o Opin não está a funcionar bem, podem ir à sala de suporte técnico que está nas sessões e podem esclarecer qualquer dúvida. Portanto, eu recomendo a todos os oradores que têm comunicações orais que deem lá um pulinho, que vão lá a experimentar como é que fazem as partilhas antes da vossa, não colado à vossa comunicação, mas mais cedo, ok? Depois, nesse menu das sessões, tem também a sala de organização, onde, principalmente nos intervalos, vai estar alguém da organização e pode retirar dúvidas que possam surgir, em termos dos certificados, em termos do programa, alguma mudança necessária. Portanto, podem encontrar-nos lá, principalmente nos intervalos. Além disso, se andarem para baixo aí nas sessões, e vocês podem ter várias janelinhas abertas e ver o Opin em várias janelas, nas sessões tem também as salas dos insetos. A sala abelha, a sala libélula, a sala vespa, e aí é onde vão decorrer os debates informais, que já anunciamos 2, mas podem propor mais debates informais, portanto, sítios onde fazem brainstorming sobre determinados temas, contactem-nos e podemos agendá-los. E também podem usar essas salas para conversar com os vossos amigos, os vossos colegas, quem quiserem. Portanto, é só entrarem dentro das salas e usem-nas à vontade. Quem tiver vontade de mais dinamismo, podem usar o espaço de networking e aí têm tipo speed datings curtinhos com pessoas ao acaso para falar sobre os temas que quiserem. Portanto, eu também vos desafio, assim, numa lógica mais arrojada, a experimentar. E depois, no espaço das exposições, no finzinho, em baixo, têm os pósteres todos e têm mais alguma informação sobre os patrocinadores. Portanto, nesse espaço de exposições, basta usarem o menu do lado esquerdo em cima, no topo em cima, esquerdo, tem um campo de pesquisa, basta porem uma das palavras que vêm no título do póster ou no autor do póster e que está no vosso programa e conseguem encontrar os pósteres. Ou então, podem usá-los fazendo pesquisa pelos menus que estão aí, pelos temas ou pelos dias. Portanto, podem fazer a pesquisa como quiserem, ok? Portanto, usem muito o Open, explorem ao máximo. Se virem do lado direito, têm pessoas e podem clicar nos nomes das pessoas e podem falar com elas. Falar ao vivo, em videoconferência, ou mandar mensagens. Portanto, explorem ao máximo o que o Open tem para vos dar. Além disso, e antes de passar a palavra à moderadora desta sessão plenária, queria só agradecer, deixar aqui um agradecimento expresso aos nossos patrocinadores, e vou fazer como o João fez ontem, vou nomeá-los, porque eu acho que mesmo que sem eles teria sido impossível tornar este congresso gratuito, e termos acesso a esta plataforma e termos conseguido pôr tudo a funcionar. E, portanto, vocês podem, obviamente, ver os patrocinadores logo no sítio de exposição, no topo do menu, mas eu vou nomeá-los só como uma forma também de agradecimento à sua colaboração. Portanto, nós tivemos o patrocínio da Singenta, da Redes, do Raiz, do Instituto de Investigação Raiz, da Altri Florestal, do Centro Pinos, da NHBS, da Biosani, da Belchim, e depois temos ainda a colaboração da Insects, uma revista que depois vamos falar no fim, que permitirá a publicação dos trabalhos dos autores que assim o considerarem, e da Portugal Bugs, que está aqui, e que depois também temos uma surpresa para vocês, que a Portugal Bugs está a oferecer. O resto dos logos que vêm aí na participação, na entrada, são da organização. Ok, e eu acho que são aqui as notas principais. Já fiz assim um bocadinho a visita guiada aqui só para começarmos a entrar dentro da lógica de Open. E vou passar a palavra à Carla Rego para fazer a apresentação da plenária. Obrigada, Carla.
Muito obrigada, Elia. Good morning, everyone. It is a pleasure to welcome Thomas Wood in our Iberian Congress of Entomology. Thomas is a postdoctoral fellow at the University of Mons in Belgium, working on ecology and taxonomy of wild bees, particularly on the highly diverse genus Andrena. He has studied the bee fauna of several countries, but is particularly interested in the Western Mediterranean region. We are lucky that he is. During the last years, Thomas has made significant contributions to the knowledge on Iberian bees by describing several new species to science, reporting many data on species distribution and ecology, and providing species lists and catalogs, of which I want to highlight the book on the bees of Portugal. Thomas Wood has not only contributed directly with his publications and research work to better knowledge of the Iberian bee fauna, but he has been committed to make public the important ecological role of bees and also stimulate young researchers on the study and conservation of this important group of pollinators. With this goal in mind, Thomas led a very successful course on identification of bees in Lisbon 2 years ago. He made significant contributions for the red listing of bee species in Portugal, and more recently joined us during the World Bee Day to make public and commemorate bee diversity. Thomas will now present us with his talk on revising the bee genus Andrena in Iberia, progress and changes. Thank you for accepting our invitation, Thomas, and you have the floor.
Well, thank you very much for inviting me. So I'm going to share my presentation, and hopefully everybody can see this in full screen. So yes, I would like to talk about my favorite genus of bees, the Andrena, and the progress and challenges that I am facing in revising.
Thomas, I apologize. We only see you. You're not sharing your screen.
Okay, let me try sharing directly. Is that any better?
No, still only you. Sorry, now we see the presentation, yes.
Oh, okay. So you can still see the presentation?
Yes.
Okay.
Thank you.
Great. So yes, let's get started. So what are the Andrina? The Andrina are very, I would say, nondescript bees. They're not particularly flashy or showy. They just look generally, in my opinion, like the fairly archetypical bee. They are an extremely diverse genus. There are currently described over 1,600 species, second only to Lassiglossum. They are a relatively young genus of bees. Bees as a group evolved 110 to 140 million years ago in the Cretaceous period. Andrina are only around 25 million years old, so comparatively young. They speciate extremely rapidly, and they contain a mixture of pollen specialists, species that will collect pollen from a single botanical family, and generalists that will collect from many, many different families. So they have the ability to colonize many different niches and display a very wide range of strategies. And as we go through, you will see lots of pictures, and they all have the same general body plan. They're all fairly compact, and they're generally very abundant in Mediterranean regions. Because of this high rate of speciation, they pose a lot of taxonomic problems. Because they are evolving very quickly, it can be difficult to separate variation from genuine interspecies differences. The best way to recognize them is in the females to look for what are called fovea, which, indicated by these arrows, are little channels alongside the inner margin of the eye that are filled with velvety hairs. So hopefully this is visible in the presentation, but this is the best way to recognize them under the microscope. And these fovea are often indicative of species differences. This is a species described, the top level, species described from Morocco. If you look at the fovea, you can see that in the species at the top, it's removed from the inner margin of the eye, whereas on the species at the bottom, it's much closer to the inner margin of the eye. So these are the kind of subtle differences that Andrina taxonomists must learn. In terms of their biogeography, they are predominantly found throughout the Holoarctic, so the Nearctic and the Palearctic. They extend south to Panama, but are not known from South America. They generally extend into North Africa, the North African deserts, where they are very species rich, but are species poor south of the Sahara. There are some species in the East African mountains and in South Africa, but generally very few species. And the same pattern is repeated in Southeast Asia, with some present in mountains south of the Himalayas, such as in the Malay Peninsula, but they are not known from Australasia. So generally, they are in the Holoarctic. Given how many species I am describing and other authors are describing at the moment, it's highly likely there are more than 2,000 species, and in the West Palearctic, probably more than 800. So there's a lot of work to do. In terms of their origin, it is a very good time to work on Andrina because there are two phylogenies of Andrina and Andrinidae that are coming out very soon, before the end of the year, that have illuminated and resolved many of the problems surrounding this group. If we look at this phylogenetic tree, we can see in the very top row in yellow, we have a neotropical group, and then we come down a little bit, and in blue, with the Ancyllandrina and the Megandrina and the Locandrina, these are the sister to the Andrina group, and these are found in predominantly in North America, And then we come down a little bit to where we see the first number 1. These are the Cubiandrina that were historically considered to be a group of Andrina. They are found mainly in Turkey and the Middle East. They are actually a distinct genus. So we can move the Cubiandrina out. So we come down. Hopefully you can see my mouse. We come down here to about 25 million years ago, where we see number 2 branch off. This is when Andrina probably arose, about 25 million years ago. And these oldest groups are predominantly found in the East. So if we look at Melatoides and Andrina Bitsinski, these are found in Turkey and Israel. So it's highly likely the genus evolved in Turkey and the Middle East. So in Iberia, we are not at the center of diversity or the biogeographical origin, but we are close to it and connected to it by the Mediterranean basin. And so there's the origin. And so we have several groups which are poorly understood, such as Andrina relata group, which has about 7 species in Iberia, and species like Andrina aluardi. So there are several interesting basal groups in Andrina because this continues down for several pages. So these might not look basal, but they're fairly basal. So it is an interesting place to study the genus. So in terms of the peninsula, there's around 210 species. I can't give a more concrete number at the moment because it's still somewhat in flux because of the difficult species groups you will see as we go through the presentation. This is a bit under half of the species known from Europe and around a third of the species in the West Palearctic. And it's a little bit behind Turkey, which is, as I said, the center of diversity, and Greece. But pretty good faunal richness. The last major revision was made in 1975 and 1976 in 2 parts by Klaus Warnke. But since then, there has been no full faunal revision. And understandably, the taxonomy has changed substantially in the almost 50 years since its publication. So Warnke listed 178 species, but many more have been added since then. And more species have been described, and our understanding has changed. So I'm currently critically revising the list. I'm working on cryptic species, and I'm describing species that Warnke simply did not examine material of. We will discuss why he may not have examined material of them, and species that will just undiscovered. So in terms of the affinities, so Iberia obviously has many biogeographical affinities, which explain the large number of species present. So I wanted to cover a few of these, mainly continental steppic affinities, Atlantic versus Mediterranean tensions, Europe versus North Africa, and isolated mountain systems. So this is a distributional map for a subgenus called Parandronella. And the details are not important, but what it represents is it represents a very typical pattern of what I would call steppic species. So this group are generally found in open, dry grassland habitats. And you can clearly see there is an eastern bias. This group goes throughout Eastern Europe, Turkey, the Middle East, over into Central Asia, with one isolated species in Tunisia and actually in Morocco as well, as we have found material, and then one single isolated endemic species in central Spain. And you can see the same pattern with many bee groups. This is a species called Andrina soror, which is found in central Spain, in the high Atlas Mountains, in steppic grassland, and in the steppe grassland of eastern Turkey. So this disjunct distribution, or this eastern distribution with remnant species in Iberia, is very typical. You find them I was lucky enough to spend 3 months in Spain this spring and summer collecting, and you find Andrena sorrel in this kind of cold steppe remnant, which is increasingly being taken over by olive groves, unfortunately. Okay, the Atlantic mountains of Spain and Portugal also hold an interesting and endemic fauna. And I've highlighted the most interesting area from the perspective of Andrena. So there's a different subgenus called Andrena tiny Andrena, which has a misleading pronunciation because they are not tiny, they're not very small, they're medium-sized. They are very strongly associated with the family Fabaceae, as you can see here. So one of the problems with one of the features of Warnke's work is that he often had a very broad interpretation of species, because when you work at a West Palearctic scale, you see enormous variation. And so he often described or created many subspecies where there was some difference, but he did not feel it was sufficiently large to have a valid species. So this is a distribution map of a species called Andrena wilkella, which, as you can see, is very widespread, found across Europe. And then in Iberia, marked with the letter B, is Andrena wilkella beaumonti, which Warnke believed existed in central Spain and central Portugal. It was described, if you look at the single dot in Morocco, it was described from the high Atlas Mountains. So here we have a picture of Morocco, and this is the locus typicus of Andrena beaumonti. So clearly very isolated from Iberian populations. To have a distribution of the Sistema Central and the high Atlas is quite isolated. So I was able to collect specimens of this putative subspecies from this very Atlantic kind of habitat in northern Portugal, in Minho province. And if you compare the Iberian and the Moroccan, you do start to see what, as an Andrena taxonomist, I consider meaningful variation, such as the thickness and the density of the pilosity, the hairbands. You can see on Tergite 2 that there is a gap, whereas it's almost complete in the Moroccan material. But much more clearly in the males, you can see, again, it may seem to be a subtle difference, but as an Andrena taxonomist, you can see that the gonocoxa here are touching, whereas they are separated, and the form of the gonostyle is more elongate, and the penis valve is slightly narrower. So when we back this up with molecular data, this is a molecular phylogeny of Iberian members of the subgenus. If we look up here in the top half of the tree, you can see marked in green, we have the true Andrena wilkella. And if we look in blue, we can see that actually, or if we look first at the top, Andrena beaumonti, the Moroccan material, is actually genetically differentiated from wilkella. And if we look in blue, we can see actually the Iberian material is differentiated both from the material in Morocco and the material in Central Europe. So we actually do not have one single widespread species, we actually have three species. One found across Europe, one found in the mountains of central Spain, and one found in Morocco. And this map gives you an idea of the way these species overlap. If we look first in black, we can see the true Andrena wilkella, which is isolated to the Pyrenees, the Cantabrians, and then comes down into northern Portugal. We can see in blue the new Andrena benwasti. So it can be found in sympatry with Andrena wilkella, but the genetics suggest, and the morphology suggests, that it is valid and distinct. In red, we just have a different overlapping species, which should be treated as a valid species in this same group. And in green, we have a completely overlooked species, which we called Andrina levanti. Warnke, in his revisions, examined predominantly material from Dusmet, which was housed, or is housed, at the museum in Madrid, because Dusmet collected predominantly in central Spain. Not entirely, but predominantly. And so we have good coverage of taxa present in central Iberia, but relatively little material from southern mountains. And so this is where we are finding completely overlooked species. And again, when we look in central Spain, in the Sistema Central, we're able to see the kind of habitat in which Andrina benwasti, this new species, exists. This very interesting mixture of Atlantic and continental affinities. Yes, including mixtures of species such as Andrina elata, which is completely endemic to Spain. But it also contains these more Atlantic habitats where you can find species such as Andrina saxonica, which is a Central European species, but it extends into central Spain in these Atlantic habitats. So that influence, that Atlantic influence, continues to support an endemic and an Atlantic and a Central European fauna, all mixed together. This is again well illustrated. This is a species called Andrina angustior, which is found within the subgenus Euandrina. This species was described from England and is very common in Northern Europe in Atlantic habitats. And this is what was described as a subspecies, Andrina angustior impressa, described by Warnke. But there is morphological variation. And he thought, well, it's similar, let's call it a subspecies. And you can look again at one of his distribution maps. Marked in A, we have the Atlantic angustior. Marked in B, we have a species called fulvata. And marked in C, in the bottom, in the peninsula, and in North Africa, we have impressa. And he believed that there was just one single species with some kind of variation as you proceeded north to south. But as an Andrina taxonomist working today, this kind of variation is meaningful. We see a difference in the degree of chagrination on the tergal margins, a degree in the coloration of the terminal fringe. So it's extremely likely that they are valid species. And of course, when we do the genetics, you can actually see it's not even close. Here we have Andrina fulvata, the continental form. Here we have angustior in blue, the Atlantic form. And we can see here at 1551, we have a specimen from Portugal, which clearly falls into the northern group. And higher up, we have impressa from Portugal, from the same area, clearly separated. So very, very strong evidence that we have two species, or three species in this case. And here is an updated distribution map. We can see that Warnke was broadly correct. If we look in red, in Northern Europe, we can see we have Andrina angustior. In green, we have Andrina fulvata. And in blue, we have Andrina impressa. But we also have Andrina angustior present in those Atlantic parts of Iberia. In some cases, overlapping completely with impressa. And another species I was able to find, if you go to the Sierra Nevada, in southern Spain, you can find impressa very commonly, where it will favor Mediterranean shrubs, particularly Scrophularia. So if you are in southern Iberia or in central or northern, keep an eye out on Scrophularia because you're likely to find Andrina impressa, which will favor herbs and shrubs, whereas Angustia will favor Atlantic scrub habitat rather than Mediterranean. So you can see there's not only a genetic difference, there's also an ecological difference. The different affinities of Iberia allow these species to exist very close to each other. Okay. Another reason that Warnke didn't describe some of these species is because they are found in extremely restricted habitats. This is a species I described this year, called Andrina juliana from San Julian in Malaga. And I described it from two specimens collected in 1962 and 1986. Both of these localities were very close to saline estuaries, close to the sea. And I suspected that they may be halophiles. They may be associated with sandy, salty soil. But I had no direct evidence. Amazingly, I was able to find this species in Malaga province in saline lagoons, although in this case, a dry saline lagoon, because by June it was already quite hot. And on the very edge, close to, you see there is tamarisk vegetation. You can find the species in abundance. This is Andrina juliana. In fact, you could catch thousands of them if you wanted. And the same was also true in Murcia province, where, again, on inland saline salt marsh soils, I was able to find this species in abundance. So if you have strange habitat that maybe you think is not very good for bees, always investigate, because this species was hidden because it lives in these strange undersampled places. So always ABC, always be collecting. Okay, outstanding problems. There's a subgenus called Micrandrina, which is basically the very small black andrina that everybody hates because they are extremely difficult to identify. There are around 30 species in Iberia, and very difficult to identify. I was able to sample in the Sierra Nevada, and when you get to high altitude, you find lots of Micrandrina. There's this wonderful little pink brassica, and this species or subspecies was described by Warnke as Andrina niveata lacana. I am currently doing genetic work, but it's likely that this will be elevated to full species. So to resolve all of the problems in Micrandrina, it is absolutely necessary to do genetic barcoding. We see similar problems away from the Sierra Nevada. This is in the Sistema Iberico in Teruel province, where you have these amazing dry valleys, very high at nearly 2,000 meters. And you can find many Micrandrina collecting on sedum, Crassulaceae. This is a very good plant to search for small andrina, and work is ongoing as to whether this is an undescribed species or not, but I cannot comment further until I have barcodes. It's reminiscent of a problem in Atlantic Iberia in the northwest. I described a species called Andrina omnilevis, which collects pollen exclusively from sedum, but it's found in acidic Atlantic mountains in the northwest, which are very, very dry, different to the dry mountains of the Sistema Iberico. So lots of questions and lots of very subtle variation. So I hope to resolve these problems soon. And then again, the subgenus Tiny Andrina, as we saw from the Wilkella Benwasti Beaumonti problem, there are many cryptic species that have been grouped together, and mountain species have been overlooked. And this same habitat at the Refugio La Campinuela, we also have more undescribed endemic species, this time a species of Tiny Andrina found only in the Sierra Nevada. So there is a lot of work to do this winter describing these new species. And the Andrena avacula complex is a complete mess, and across Europe it contains at least 2 species. Both of these species are found in Iberia. So if you have tiny Andrena, put them on one side and wait a year until I have revised this group, because the number of species in Europe will increase from 11 to more than 20. So there are very many unanswered questions. So in conclusion, I hope I've introduced you to this genus and to the problems that we currently face. But it is a tremendously rewarding group to work on, especially in Iberia, because the very large number of biogeographical affinities that exist mean you have many of these different species found close together. If you work only in Central Europe, you often only see one of the species, and so you cannot see the variation. But in Iberia, you often have multiple cryptic species close together. And this means that we can see there are 2 things, not just one thing. We can see this in the Angustial group. We can see it in the tiny Andrena group and the UN, yes. So always go to these strange overlooked habitats. Go away from central Spain. Varnca covered central Spain. It's the southern mountains, the northern mountains, the salt marshes. If you see a strange plant, if you think this plant is no good for bees, always look, because you may be surprised. We've had 7 endemic Iberian Andrena described in the last 2 years. I am sure there are many more, so keep looking. All of this work will be brought together in the publication of a key. I hope that this key will be ready and published by the end of next year. But if people send me new species, it may take a bit longer. So hopefully we will very quickly make this key obsolete. But yes, thank you very much for listening, and I'd be very happy to take any questions you may have.
Tom, for this fascinating talk. It's always a pleasure to hear you talk about this diversity. It's under our eyes, but it's so underestimated. We have a question from the audience. I apologize beforehand on my lack of good pronunciation in Spanish names, but it's from Miguel Lopez Manguina that asks you, what kind of genes are you using in your genetic analysis?
Yes, so I am a very simple taxonomist when it comes to genetics, and Andrena genetic taxonomy is still quite young. So we are only using the CO1 fragment, the cytochrome oxidase 1, which is the standard barcode fragment. We mostly use the LEP forward and reverse primers. Sometimes we use a subset. Because Andrena evolved very, very quickly, they are often very difficult to barcode, probably because there are mutations at the binding site for these primers. So sometimes we have to use more primers designed for the different groups of Andrena, but they are always amplifying the same CO1 fragment. We are not at the position of bumblebees, where they can use PEP and other markers. So just straightforward CO1.
Okay. And from Ricardo Cardoso, does each species of Andrena have a main family of plants to feed on, or is it present only in certain species of Andrena? if, for example, certain species are genetic and others prefer just a plant family? Generic, sorry.
Yes, so my current fellowship. So I'm currently working on a fellowship, and this is one of the broad questions that I'm working on. So, does each species of Andrena have a main family of plants? So there are many species of Andrena that are strict specialists. So, for example, the tiny Andrena that I showed you, the Wilkella, Beaumonti, Benoisti, they are pretty much all specialists on Fabaceae. But if we look in other lineages, such as the Melandrena, that includes species like Andrena flavipes, which is very common in Iberia, they are very strong generalists. So if we look across the phylogeny, there's a complete mixture of specialists and generalists. It's very difficult to see a consistent pattern. I think there is a pattern, but it needs a lot of work on the basal clades. Generally, if you look at the oldest groups of Andrena, in my opinion, they are more specialized, whereas the very young groups of Andrena are more generalized. But a lot of work is needed to make this pattern clear.
Okay. And another question from Ricardo Costa. Do you see a trend of more generalist species in Central North Europe that might have quickly spread over the retreat of the ice in the last 10,000 years, or mostly specialists that move upwards?
Yeah, so this is a great question. And this is partly what my fellowship will be about. So maybe in 2 years, I will write a paper on this. In my opinion, yes. The northern fauna is more generalized, whereas the southern fauna is more specialized. The mechanism is not clear to me, however, because let's say your question is the hypothesis. Maybe they were all mixed together in Southern Europe and the generalists spread out faster. Yes, that's a very good hypothesis. The problem is that many of the specialists in Iberia are specialists on plant groups that are not found in Northern Europe. So, for example, cistus. There's a very large cistus feeding and specialized fauna in Iberia with things like Fibrella, Gelfensis, Villipes. They can't leave Iberia because cistus is not found in Northern Europe. So I think the core of your question is probably correct, but I need to work carefully on the mechanisms. But generally, yeah, the generalists go to the north much faster.
So we keep the specialists down here to fascinate.
Yeah, I mean, it doesn't work. It doesn't work for every group, because if you look at the Andrena sensu strictu, which is more diverse in Northern Europe, because they are mainly feeding on trees, so or salix, willows. So those species are actually very rare in Iberia because there is comparatively little deciduous woodland. Whereas obviously in Northern Europe, there's lots of deciduous woodland. So it's a complicated picture. When you have 200 species in your group in Iberia, the picture is complicated. But I think the core is probably correct, the core of the question.
Okay. Elia Marchant is thanking you for the very interesting talk. And is stating that unfortunately young entomologists, taxonomists are becoming rare. What would you say to young scientists to motivate them to do this type of work?
Yeah, it's a really substantial problem. In fact, in bees, I think we're probably comparatively quite lucky that we do have some young taxonomists. It's mycologists, people working in fungi, that we have an enormous lack of young researchers. In terms of working on bees, what I would say is that if in terms of motivation, the work itself is the reward. Because you're peeling back layers of complexity. You know, we work and exist and live in this extraordinarily complex world. And when you work as a taxonomist, you are uncovering complexity. You are uncovering the secrets of the world. So my encouragement is that if you want to see further and you want to see deeper, that's why we do the work. I didn't start out intending to be a taxonomist. I wanted to be an ecologist. I am an ecologist, but I wanted to be an ecologist. And when I went to Iberia, I couldn't do ecology. I couldn't do it fully because I didn't understand the species and I didn't understand the diversity. So I became a taxonomist to be a better ecologist. So that's what I would say. It helps me see more clearly.
Yeah, indeed. From Paulo Borges. With this complexity in this genus, is it possible to identify the several species in classic ecological pollination studies?
So I guess I would say two things. I would say, one, it is possible because I can do it, not for every group. You can't do it for the Microandrina, but you could do it for a lot of them. But I also have a really, really good knowledge of the genus. What I would say is that with care, so if you have a system, first you catch all the species and then you learn them. And when you've done that, yes, you can do them in the field, but it requires having a knowledge. You first must have a microscope knowledge, and then you can have a field knowledge. But it can be done. Andrina flavipes, Andrina nigerania, like you can learn a lot of the big ones. But you need first to learn them under the microscope. And I hope that with the key, when it is published, people can learn them under the microscope and then take that knowledge to the field.
This is from me, just a question related with this topic. What is your feeling about doing what about the keys for dummies for bee identification? That means that many people are interested in identification bees for their own pleasure or just because they are working in pollination, for instance, in different sectors of activity. And what do you feel about academics, taxonomists producing or helping to produce more easily used keys, mainly using pictures, for instance, whenever it is possible, where the features are more easily observed by people not so familiar with the microscope work and not so trained?
Yeah, so it's an interesting question. So just yesterday, we had a meeting, the first meeting for a project called Orbit, which has just started at a European level and is aimed at, it's funded by the European Commission and is aimed at producing identification guides and resources for all European bee species. And it's going to take the form of fact sheets for each species with photographs and identification characters. So I think this is an important resource. In the context of Andrina, when you have 200 species, you've got to be so careful about giving people too much confidence. Okay. If I produce a guide which says, here's a photograph of an Andrina, and you look here and here and here, people might, you don't have in your head the concept of all the other variation. Mm-hmm. So people will look at it and go, well, my bee looks kind of similar. So I think that can be done, but the gold standard will always be a full key.
Of course.
So things will happen in terms of photographic identification. Mm-hmm. but I will still produce a real key. That's the gold standard.
Okay. I think I have two questions. Elliot, can I make them? You have your microphone turned off.
You're muted.
Sorry, I was saying that we are short on time. I would challenge the last person to contact Thomas Wood directly, and I'm sure he will be pleased to answer. Because I guess you can select a last one, but then we are taking time from the sessions, okay?
Okay. So I apologize for the one left behind, but I'm going to choose the one from Rita Moraes. It has a follow-up question on the Ricardo's above that we did earlier. Are we to expect that these younger species will also specialize, or will they remain generalists? Will this depend on where they are found?
So very quickly, as a rule, we expect the young species to generalize. Yes, it depends on where they are found. If you look in the very young clades of Andrena, you mostly find generalists, but you do also find some very young specialists. For example, in Greece, you find groups that specialize on crocus, which are very young. So yes, mostly generalists, but yes, there's also context important. Context is important. So you learn very quickly in Andrena. It's hard to make strong rules that always apply.
Indeed, with such an amount of diversity, it's always hard to put patterns in place.
Yes.
So thank you, Thomas, for your fascinating talk. Thank you to our audience for our very interesting questions. I apologize for João Cardoso. I will post your question to Thomas in a few moments, so we can answer you privately. And I give the floor to Elia to make some announcements.
Okay. Thank you, Thomas, also. It was very, very nice. Só quero lembrar-vos, por favor, de registarem-se no BioBlitz. Continua a haver aqui muitos nomes em falta, ok? Se tiverem dúvidas como é que podem fazer, nós vamos postar aqui nas notas do Opin. E além disso, a partir de agora, têm um breve tempo para ir tomar um café e esticar as pernas, mas tentem entrar. Quem tem sessões, os oradores das sessões agora já a seguir, eu diria para entrarem já na sua sessão, portanto já estão as salas criadas neste momento nas sessões, escolham a vossa, entrem e já lá estarão os moderadores à vossa espera. Até já!